Okay, so welcome everyone to this very first session of the Biodiversity Genomics 2024 conference. Here, our session today focused on ERGA, the European Reference Genome Atlas. We only have just less than an hour together today, so it'd be absolutely impossible for us to tell you absolutely everything that's going on inside ERGA within this short time that we have together today. But we picked a few highlights, if you like, to try to explain, especially to newcomers or first-time people interacting with ERGA, what ERGA is about, some of our activities and some of our outputs. So we have a couple of speakers lined up for today. Firstly, Chiara Bortolucci will be presenting Navigating the European Reference Genome Atlas. She will be followed by Astrid on the sampling side for reference genomes through the community efforts. Then we move more on to the kind of data management side of things with a presentation from Tyler on the ERGA genome tracking console. And then Diego will walk us through some of the latest efforts in building this decentralized approach, if you like, for the community reviewing of genome assemblies. And finally, hopefully there'll be enough time at the end for me to briefly introduce you to our concept of ERGA community genomes and leaving possibly 10 minutes at the end for some open Q&A session. So firstly, just an overview of, especially for the newcomers here, on what is ERGA. So the European Reference Genome Atlas is the European node of the Earth Biogenome Project, which many of you will have heard of, this moonshot mission, if you like, for biology, aiming to sequence, catalog, and characterize the genomes of all Earth's eukaryotic biodiversity, with this grand vision for establishing a foundation for biology, aiming to preserve and restore biodiversity and in a One Health approach to looking after humanity as well. And this Earth Biogenome Project is, of course, a network of networks, regional nodes being, for example, the African Biogenome Project, and then us in Europe, the European Reference Genome Atlas, but also taxonomic-focused projects like the Vertebrate Genomes Project or geographic-focused projects like the Darwin Tree of Life in the British Isles. In ERGA, you will have possibly heard of the different committees that are supporting all of the different aspects of the reference genome generation workflow. And this involves all the major steps along the way, from the sampling side of things through to the sequencing, into the assembly, followed by genome annotation, and with the downstream data analysis. And of course, this all needs to be supported by IT and infrastructure for managing all of these different steps. But we also take the broader view of making sure to also consider things like ethical, legal, and social issues, media, so how we communicate the importance of genomics and reference genomes to non-specialists. The Citizen Science Committee is also engaged with taking that message to the amateur scientist, if you like, who is interested in contributing to biodiversity, but maybe has less of an understanding of genomics. Then through the Training and Knowledge Transfer Committee, of course, that's incredibly important, especially for our early career researchers, to give them the confidence to be able to perform all of these different steps of the reference genome generation workflow. And finally, our newest committee, the Social Justice Committee, making sure that this is done in an equitable way. In terms of our governance structure rather than our operations, one aspect that is very important to ERGA is this representation across the European continent. So no matter the level of maturity of any country's biodiversity genomics efforts currently, we wish them to be part of this process so that we all walk together towards being in a position in a few years' time where being able to generate high quality reference genomes for the entirety of European eukaryotic biodiversity is possible. So that's what I'm going to say in terms of an introduction, and then I can hand over to Chiara for the first main talk of today.
Thank you so much.